Hey guys, we are going to start taking a look at the chapter 11, which has to do with genetics. And as you see on the screen, the work of Gregor Mendel. So, what is inheritance? It is something that we receive from our parents, a contribution that determines our blood type, our hair color, and so many more things. So, what kind of inheritance makes a person's face round or their hair curly? These are all the different traits that we're going to kind of talk about as we go through this. So the experiments of Gregor Mendel. Mendel is kind of considered the father of modern genetics. And some of the questions that he looked at are: where does an organism get its unique characteristics? An individual's characteristics are determined by factors that are passed from one parental generation to the next. Every living thing, plant or animal, microbe or human being, has a set of characteristics inherited from its parent or parents. Delivery of characteristics from parent to offspring is called heredity. So there's a difference between heredity and genetics. So the scientific study of heredity, known as genetics, is the key to understanding what makes each organism unique. So Mendel was an Austrian monk. And he basically, he wasn't a scientist per se. He basically was the guy in charge of the garden, and that's where he did all of his work. So the work that he did there ended up being hugely important. Mendel carried out his work with ordinary garden peas, partly because peas are small and easy to grow. A single pea plant can produce hundreds of offspring. So today we call peas a model system. And that's one of the reasons why he was so successful in doing this, was because he just happened to pick something that gave him very, very good results. By using peas, Mendel was able to carry out in just 1 or 2 growing seasons experiments that would have been impossible to do with humans and would have taken decades, if not centuries, with larger animals. But because peas have a short generation time and they're very easy to manage, he was able to get a lot of data in a relatively short amount of time. So Mendel knew that the male part of each flower makes the pollen, which contains the sperm. So that's what pollen is. Pollen is plant sperm. And you can see in the picture the male parts and then the female parts. So the male parts are around the sides, and then the female part is in the middle. So similarly, Mendel knew that the female portion of each flower produces cells called eggs. So eggs are created in the female part. During sexual reproduction, male and female reproductive cells join in a process known as fertilization. I don't think this is anything you guys haven't heard before. But so when you get a sperm and an egg and they come together, they fertilize and they make an embryo. In peas, this new cell develops into a tiny embryo encased within a seed. Pea flowers are normally self-pollinating, which means that sperm cells fertilize egg cells within the same flower. So normally, one flower will fertilize itself, which leads to making genetically identical plants. So a plant grown from a seed producing self-pollination inherits all of its characteristics from a single plant. In effect, it has a single parent. Mendel's garden had several stocks of pea plants that were true breeding, meaning they were self-pollinating and would produce offspring with identical traits to themselves. In other words, the traits of each successive generation would be the same. A trait is a specific characteristics of an individual, such as seed color or plant height, and may vary from one individual to another. He took a look at a whole bunch of different traits, and we'll talk about some of them in here. So this is basically how he did it. So Mendel decided to cross his stocks of true breeding plants. He caused one plant to reproduce with another, and you can see that he basically took kind of like a paintbrush and took the pollen from one plant and then crossed it over to the other plant, and then was able to fertilize plants that were not the same in that way, even though normally they pollinated themselves. To do this, he had to prevent self-pollination. He did so by cutting away the pollen-bearing male parts of a flower and then dusting the pollen from a different plant onto the female part of the flower, as shown below. So notice over here, the plant on the right, he cut away the male parts, and that way he was absolutely sure that this plant did not self-fertilize. This process, known as cross-pollination, produces a plant that has two different parents. Cross-pollination allowed Mendel to breed plants with traits different from those of their parents, and then study the results. Mendel studied 7 different traits of pea plants, each of which had 2 contrasting characteristics, such as green seed color or yellow seed color. Mendel crossed plants with each of the 7 contrasting characteristics and then studied the offspring. The offspring of crosses between parents with different traits are called hybrids, and you'll get a sense of what that is coming up here shortly. So here are the 7 different characteristics, and just a little bit of vocab here. So this is the parental, this P generation is the parental generation. So these are the 2 that are being crossed, and this is the F1. That is the first filial generation. So these are the offspring. So these are the 7 different characteristics that he looked at were round versus wrinkled peas, yellow versus green peas, gray versus white seed coat, smooth or constricted pod shape, green or yellow pod color, axial versus terminal flower position. So axial would be that it was closer to the stem. Terminal would mean that it was as far away from the stem as possible. And then tall versus short. When he did this, he noticed that whenever he crossed these 2, he got a round. Whenever he crossed these 2, yellow and green, he got yellow. Gray and white, he got gray. Smooth and constricted, he always got smooth. Green and yellow, he always got green. Axial and terminal, he always got axial. And tall and short always gave him tall. As I said before, the F1 generation, this is the first offspring, so the first filial generation. So for each trait studied in Mendel's experiments, all the offspring had the characteristics of only one of their parents, as shown in the table, and that's what I had just gone over. In each cross, the nature of the other parent with regard to each trait seemed to have disappeared. So none of the traits that are listed on the bottom here showed up at all. So he kind of wondered how and why that would happen. From these results, Mendel drew 2 conclusions. His first conclusion formed the basis of our current understanding of inheritance. An individual's characteristics are determined by factors that are passed from one generation to the next. Scientists call the factors that are passed from parent to offspring genes. Each of the traits Mendel studied was controlled by one gene that occurred in 2 contrasting varieties. These gene variations produce different expressions of or forms of each trait. The different forms of a gene are called alleles. So the alleles here would have been like tall versus short, green versus yellow. Those are different alleles for the same trait. In this chapter, you really kind of have to keep track of the vocab because there are a lot of different words that you guys probably haven't used before. Mendel's second conclusion is called the principle of dominance. This principle states that some alleles are dominant and others are recessive. An organism with at least one dominant allele for a particular form of a trait will exhibit that form of the trait. An organism with a recessive allele for a particular form of a trait will exhibit the form when the dominant allele for the trait is not present. So if you look down here, this told him which trait was dominant. So all the ones down here that showed up in the F1 generation, these are all the dominant alleles. So how are different forms of gene distributed to offspring? During gamete formation, the alleles for each gene segregate from each other, so that each gamete carries only one allele for each gene. So Mendel wanted to find out what happened to the recessive allele. So he took 2 hybrids. A hybrid would be an organism that had both, say in this case, a tall and a short parent. So here's the tall and the short parent. Here's the F1 generation, which both look tall, but he knew that they had a short parent. So then he took these plants and he crossed them, and his results were down here. He got 3 tall and then 1 short, which meant that the allele, the short allele that came from the parent up at the top in the parental generation, did in fact make it through and ended up being shown down here. So when Mendel compared the F2 plants, he discovered the traits controlled by recessive alleles reappeared in the second generation. So that's this one down here. Roughly 1/4 of the F2 plants showed the trait controlled by the recessive allele. Mendel assumed that a dominant allele had masked the corresponding recessive allele in the F1 generation. The reappearance of the recessive trait in the F2 generation indicated that at some point the allele for shortness had separated from the allele for tallness. And we'll kind of get into a little bit of that later. So how did this separation or segregation occur? Mendel suggested that the alleles for tallness and shortness in F1 plants must have segregated, so separated from each other during the formation of sex cells or gametes. In other words, sperm and eggs. Your gametes are your sperm and eggs. So what this is saying is that, hey, if you have a tall and a short allele, then when you go to make your sperm or eggs, then tall is going to go on one sperm or egg, and then short is going to go on another sperm or egg. And basically, this is the picture kind of showing how that would happen. So you have the 2 talls here, but both these are hybrid. Both have a big T and a little t. Big T and a little t. And when you segregate these out, what you end up seeing is that you get the same 3-to-1 ratio. 25% end up with 2 little t's, so those would end up being short. We're going to stop there, and we're going to pick that up more next time, where we're going to talk about Punnett squares and do some problems.